Good morning. If you are watching on for parts of the world that it is morning, I would like, on behalf of the FF-IPM project partnership, I would like to thank you for your participation to our webinar entitled Fruit Fly Detection and Interception, the FF-IPM project response. We are happy to welcome you to this webinar in the frame of the FF-IPM project, a European research project for the protection of fruit production and trade threats posed by fruit flies under the Horizon 2020 European funding for research and innovation. Today, with this webinar, we are launching the FF-IPM series of webinars with interesting topics that will follow in the next months, always in the frame of the FF-IPM project, covering its scope and objectives. Before giving the floor to our moderator, Professor Papadopoulos, I would like to ask you to stay tuned because we are going to inform you about what it is to follow in the next month. So I think interesting things are coming. So you are invited to follow us and participate in the next events that we are having in our mind to plan for you the next months. Minor details that I would like to inform you about today's webinar is that this webinar is being recorded. So in order for us to have it widely disseminated for a wider audience to watch it further at some point. And you are invited to send us your questions, your thoughts, your input. It is valuable via the Q&A button at the bottom of Zoom, as you can see it. Mr. Meyer will dedicate enough time to after his presentation in order to cover your questions and thoughts. So I now think that we can start with Professor Papadopoulos, Professor of Applied Entomology, Director of the Laboratory of Entomology and Applied Zoology at University of Thessaly, who can introduce us to the topic and to our presenter. Professor Papadopoulos, thank you for your contribution, and we are ready to start.
Thank you very much, Georgia, for the nice introduction, and thanks to all our attendees and Marc the Meyer for this event today. So good morning, everybody. I'm really glad to welcome all of you in the seminar on fruit fly detection and interception. And this is actually related to the FF-IPM project. This is part of a series of webinars, as Georgia explained earlier, and you should stay tuned and follow for the next events. Our project works for the last 2.5 years on developing tools, approaches, and strategies to address invasive fruit flies in Europe. And in fact, this series of the webinars, they come also to contribute to this direction. So the event today, as I said, focuses on fruit fly detection and interception, and we all know that this is a really important topic because this is to avoid entering of new organisms and new fruit flies in the European territories. And if they accidentally arrive, then we should be able to detect them early and respond with some aggressive interventions. So I would not like to say more, but I would like to say a few words for our speaker of today, Marc the Meyer. Marc is an entomologist, an experienced entomologist in taxonomy, systematics, and phylogeny with focus on Diptera and on the group of fruit flies, on the Tephritids. Museum for Central Africa in Belgium, and currently is the head of the invertebrates unit, and it's an acting head of the Department of African Biology. So there is a list of titles for Marc, and I wouldn't like to go through all of them. But you know, Marc is the chair of the scientific organization of TEAM, The team is definitely workers of Europe, Africa, and the Middle East. It's also a member of the steering committee of the International Fruit Fly Organization. And Marc spent a long time in Africa working on fruit flies and on other subjects. He is still working in several African countries and participates in many initiatives there. He is the technical manager of the FF-IPM project and with a major contribution. So, Marc, the floor is yours.
Thank you very much, Nikos, for this nice introduction. So, welcome to everyone. This is, as Nikos and Georgia already explained, the first webinar within the Fruit Fly IPM project. So, it's me to take the flag as the first one and see how it goes. But without much ado, I will start my presentation straight away. I will therefore share my screen. Like this. I think you can see my screen at the moment.
Yes, of course.
Okay, perfect. So the topic of today is fruit fly detection and interception, the fruit fly project response. It is a part of the fruit fly IPM project with the full title In-silico boosted pest prevention and off-season focused IPM against new and emerging fruit flies. It is an EU project within the framework program Horizon 2020, and the funding is from the EU. It started in September 2019, and it is a 4-year project. The project itself is conducted by a consortium of several institutions, private companies from throughout the world, with an emphasis, of course, of European institutions, but we also have participants from African countries, from China, Australia, the United States. And so it's a fairly wide or fairly large consortium with 21 partners. We have more than 70 scientists which are active in the project, coming from 17 countries. And the main coordinator is the moderator of today, Professor Nikos Papadopoulos from the University of Thessaly, who runs the whole project. So how does this project come about? The EU realized that there are new and emerging risks to the EU plant health posed by organisms in general. And for that, they organized a call within the Horizon 2020 program. And the consortium replied to that one and said that one of the major groups that are of importance in this framework are fruit flies. After an evaluation, the project by the EU to be run as a project. Why fruit flies? Well, when we talk about fruit flies in this respect, we talk about what we call the true fruit flies, the dephritic ones. You all are probably aware or known of the Drosophila, the ones that most people know as small flies that you find in the kitchen when your fruit is overripe. These are not the topic of this research. These are the true fruit flies, the dephritic. What is the difference? Well, the fact is that dephritic fruit flies have a kind of a parasitoid life cycle. The females will deposit their eggs inside fresh fruits, usually when it's still on the tree. The larvae will develop inside the fruits and destroy thereby the interior, the fruit mesocarp. After the larvae are full grown, they will leave the fruits and they will go into the soil. There, the next stage, the pupal stage, will take place. It can take several weeks, or it can even take several months. It depends a bit on the kind of fruit fly. And then when the fly is fully grown as an adult, it will emerge from the pupa, emerge from the soil, and then the life cycle starts again. They will mate, fertilized eggs will be produced, and they will be laid again into the fruits. Most of the species of fruit flies are multivoltine. What does that mean? That they have different generations in 1 year. But in temperate regions like Europe, we also have a number of fruit flies which are univoltine. They only know 1 generation a year, and the most well-known group of that is the Ragolitis flies. So that is the general life cycle of different fruit flies. Is that a problem that is new to Europe? No. We already have a number of fruit fly pests within Europe. However, the number is limited. We have a few indigenous pests. They mainly belong to the genus Ragolitis, and they are specialized feeders. They will only attack one specific crop, one specific fruit, or a few related ones. We have specialized feeders on cherries, for example, on walnuts, and on a number of other wild fruits. However, throughout the years, we already have encountered invasive pests within Europe, and the invasive pests that are established at the moment within Europe are mainly from African origin. The 2 most important ones are the Mediterranean fruit fly, known to most people as the medfly, Ceratitis capitata. It is a species that is of African origin, probably introduced in the 19th century in the Mediterranean region from Africa, and afterwards it spread to other parts of the world. It now has established populations in Latin America and Australia, for example. We have another one that is also widely known, and that is the olive fruit fly, Bactrocera olei. Although most people think it's an indigenous species to Europe, it actually is also an invasive pest. The only thing we don't know is when exactly it entered the Mediterranean region. But the origin of the fly itself is also of an African origin. In addition to these 2 invasive pests, we have a few other ones which have been introduced from temperate region, mainly from North America, and they belong to the group of Ragolitis. So why are we facing an additional challenge regarding fruit flies? Well, first of all, we see that some of the fruit flies that are established, like Ceratitis capitata, is actually extending its range, it's extending its distribution. Although the fly has been restricted to the Mediterranean region for a long time, in the last decades, we encountered them more and more into more temperate regions north of the Mediterranean region. In the last years, we had records from central parts of France, northern Italy, Austria, and Romania. On the other hand, we also have more frequent encounters of flies which are exotic, which do not belong to the European fauna, from other regions. 2 of those ones are the ones that are shown here, Bactrocera dorsalis, the oriental fruit fly, which is of Asian origin, but which has been introduced into Africa in 2003, and in a few years' time spread throughout the whole African continent, including some of the islands in the Indian Ocean. And we have Bactrocera zonata, the peach fruit fly, which is also of Asian origin, but was introduced already into Egypt and the northern part of Africa in the 1980s, and then has been slowly spreading throughout parts of northern Africa. It also has been introduced into some of the islands in the Indian Ocean. So we have this dual aspect of flies that are already here, but spreading further, and flies that are not established in Europe, but that pose a risk. And for this, the fruit fly IPM project chose these 3 target species, Ceratitis capitata, Bactrocera dorsalis, and Bactrocera zonata, as their focus species to develop their project. So what are the main issues when we talk about fruit flies in Europe? We have this high propagule pressure, an increased number of introductions. We have the midfly Ceratitis capitata that seems to emerge as a new pest in deciduous fruit orchards because of this further well spread. We have detections of new tropical fruit flies over the last years. But then we also have a few shortcomings. There are gaps in ecological and climatic modeling. There is not a well-coordinated interception system throughout the European Union, and there's a lack of fast and aggressive response. And therefore, the vision of the fruit fly IPM project is to try to protect European horticulture and trade from the current and imminent threats posed by selected, emerging, and new frugivorous fruit flies. So just a short word about the concept of the project as such and its approach. The approach is actually dual. As Nikos already explained, we would like to prevent the entry of new flies, of new exotic flies which are not established already in Europe. And in order to do that, we need a very early detection. We have to speed up our detective methods to, as soon as the fly occurs within the European Union, we can recognize it, we can detect it, we can recognize it, and then we can take appropriate measures to eradicate it if possible. On the other hand, fruit fly IPM also is looking at control or containment of pest species. Ceratitis capitata is a good example. It's already here. You cannot eradicate it anymore, but you can try to control it. If for some reason a new exotic fruit fly species would become established in Europe, again, control to mitigate the impact of that fruit fly would be very welcome. And for that last aspect, the approach of the fruit fly IPM is slightly different from other ones, that we focus on what we call off-season IPM. We try to develop systems of integrated pest management, but management methods that you can apply when the fly is not in its peak period. These flies know a seasonal fluctuation. You find them in high numbers when the fruits are around and when the climate is very favorable, but the population will go down, the numbers will go down very much when winter comes, when the fruits are not around anymore and conditions are not so optimal. If we can find ways to target and to tackle the problem at that moment, it will be to the large benefit of the farmers, because it will mean that the number of methods and the amount of funds that you have to apply to control the fly will be lower just because the population is lower. However, my presentation today will mainly focus on the first aspect, the prevention of entry through early detection. So I will show you a number of tools and approaches that are developed or under development within this aspect. First of all, in order to understand that, we have to look at the key reasons for the fruit flies dispersions. How does it come that these flies are dispersing and that they are entering, or that the risk of them entering Europe is there? Well, there are three main reasons: climate change, massive food trade, and increased mobility. If we talk about climate change, we actually are all aware that we see a global warming event, and that areas with a very temperate climate nowadays seems to have warmer conditions during certain periods of the year. We all know of the heat waves, we all know about the number of days with snow in temperate countries going down. all that makes the temperate region of Europe more suitable for flies which are used to Mediterranean climates. The picture I'm showing here on the left bottom is actually a study from another group, also on Tephritis, but not Ceratitis, where they've shown that because of the number of days with snow and because of the warming up of higher areas in Switzerland, the natural barrier that the Alp mountains was presenting is actually reduced because you have valleys and pathways these days by which flies can disperse from the area below the Alps into the northern area. And so we see this general trend of the flies spreading because the climate becoming more suitable. Commercial trade, we all like to eat fruits, and we all like to eat the fruits the whole year through. Before, people were eating apples when it was apple season, and as soon as the apples were finished, you didn't eat an apple anymore. You waited for the next season. These days, you want to have an apple a day, and for that, we have to import them from other areas who have a different climate or a different season pattern like ours. If we are in our season that apples are not around, in the southern hemisphere, they may be around. So we bring them in from those areas. We also have more and more an appetite for exotic fruits. 20, 30, 40 years ago, finding a mango in a supermarket was nearly impossible, except if it was a very niche market or a niche shop with oriental foods, for example. Nowadays, mangoes you can find almost the whole year through in supermarkets, and that also applies to other fruits. All these exotic fruits bring with them a certain risk because within their countries of origin, you have fruit flies that are infesting these exotic fruits. By bringing in and by increasing the trade on those ones, you have the risk of bringing in these unwanted visitors together with the fruits. And then we have the increased mobility. The black and white picture shows you how people were traveling in the 1950s on flights, and when the number of lines was very limited. Flying was a luxury in those days and also for a limited number of people. These days, everyone gets on the plane and flies to all kinds of corners of the world, usually even more than once a year. We just don't restrict ourselves to going to the beach within our own country, but we like to go not just within Europe, but also outside, do more and more intercontinental flights, including a lot of exotic destinations. Unfortunately, people, when they are there, they often like the foods they eat locally, including the fruits, and they always would like to bring a few fruits with them for their mother, sister, or relatives to taste these marvelous fruits that they found there as well. Again, with this kind of mobility, you have the risk more and more of fruits traveling together with the fruit flies to other areas. So these are the main reasons, key reasons why we have this dispersion and this increased risk. So what are the tools that we are suggesting to help us in an early detection? Well, we have 4 groups: the E-nose, the E-traps, ID, identification tools, and prediction models. Let's first start about the E-nose. What is an E-nose and what is it used for? Well, if fruits are being introduced, and that is like in commercial trade, a part of it will be visually inspected. You will have inspectors who go through a percentage of the fruits that are being introduced. They will visually inspect them for puncture holes or for any damage done by other organisms and see if they can find any sign of infestation. Now, doing this is, first of all, not always very efficient because puncture holes, for example, from fruit flies are very tiny. It's not always easy to see them. It is also a very labor-intensive aspect because you need regular inspections of high numbers of fruits in order to recognize or to detect the few which may be infected. So it is not a very suitable or not a very efficient way. Now, what about, rather than looking for it, we could sniff it, we could smell whether a fruit is infested or not? Well, actually, that is possible. As you all know, if you take an apple or a peach and you smell it, it has a certain aroma. So there is a certain fruit odor that you can detect, even with our noses, which are not that sensitive, we can detect it. You can smell it and you can say, oh, this smells nice. This looks like a great fruit. Now, those fruit smells are actually a component of different chemical volatiles which are emitted by the fruit. And you can actually, the smell that is emitted, the volatiles that are emitted by a fruit will differ if you have a fruit which is not infested versus a fruit which is infested. So actually the smell will be different. We may not be able to detect it with our nose, but we can, with a chemical analysis, actually find the differences in the volatiles. Because the volatile, as I said, is a combination, is a mixture of different aspects. It's a bit like a perfume. If you have a perfume that you buy, you can read that, you know, it has a touch of this and a touch of that. So it is also an assembly of different odors. Well, with fruit, it's just the same. And like you may be able to smell the difference between a Chanel No. 5 and a Paco Rabanne perfume. Well, with chemical analysis, you can actually find the difference between an infested versus a non-infested. Of course, conditions are not always that easy because the composition of these smells will change over time. Your fruit will ripen, but the infestation, the deterioration inside the fruit will also increase. And the smells is difference between the fruits. An apple does not smell the same as a pear. And also, if both of them are infested, not necessarily they will smell the same. So the eNose development actually looked in all those aspects. What it works about is that it first samples the smell of the fruits, and you take a number of replicates, you smell between an infested and a non-infested. The tests were done on 3 groups of fruits, apples, pears, and peaches, and on the 3 fruit flies that are the target flies in this study, being the Capitata dorsalis and the Zonata fly. Once you have those volatiles captured, you can analyze them, and you can work out which components are included in that mixture of the volatiles. And then you can also quantify them, and you can put what they call a chemometric analysis. You can see how they differ through time and how they differ between the fruits and between the fruit fly species. And with that, you can, what we call, establish a fingerprint. You can actually define what are the characteristic volatiles that will be representative for an infested fruit, for example, an infested apple by Cerathitis capitata. Once you have done that, you can actually start training an apparatus, which is the actual eNose. You can train that apparatus by detecting it. And it's a bit like an image training. You give the apparatus a number of samples, you give him the fingerprint, and he can start establishing how to recognize the infested fruits. And so the ultimate idea of this study is that rather than having a visual inspection, you can actually go with one of these trained eNoses into a consignment of important fruits, and you would be able to smell whether there are any infested fruits inside, yes or no. The second tool that we are developing are e-traps and related to that one, automatic counting algorithms. One way of detecting flies is by trapping them. You can put up trap with specific attractants, and the flies, if they are around, will be attracted to the lure inside the trap. They will be trapped there, and then you can come and inspect the trap and see if any of the flies that is in your trap is an exotic one or not. This all sounds very nice, but again, it's a very labor-intensive work. In order to work very well, you have to put up a lot of traps, but even more important, you have to inspect them at a regular basis. You have to go there every week or every fortnight to check if there are any flies in it. So it's a very labor-intensive and a time-consuming activity. Now, rather than having to go to the trap, why can't we get the information from the trap sent to our computer? Is that not possible? Yes, it is. You can do it with an e-trap. What is the e-trap principle? Well, as you see here, on the left-hand side, you have a fruit fly trap. Fly is trapped in it, and actually attached to that trap, you have a camera. And the camera automatically is set to take a picture every day, every hour, every week. It's up to you. You can decide how frequent you want to do that. And because of the general mobile network that is currently available, you can actually, with a modem, have the picture sent to a centralized hub, a centralized computer, who will receive all these images from the hundreds of thousands of traps that are out there. And then, rather than having to go to the trap, you can, from your office, inspect the images and find out if there's any fly on it that looks suspicious or that is an exotic one and that is not supposed to be there. So within the Fruit Fly IPM project, we are developing two kinds of traps, what we call the open or the half-open trap models. And actually what they exist of is the usual trap system, a battery, because you need to have some kind of energy in order to have the camera working and the picture sent, a built-in camera, and then a modem for the image transmission. So by developing these kind of traps, you can actually speed up the detection much faster. Because imagine if you have a picture sent every day, that means that if today a fly enters this trap and it's an exotic one, you can actually already detect it today. If you have the normal traps up, you have to go there, and people cannot go every day. You will go there once every week or once every fortnight. So you actually speed up the detection at a much higher rate than by the conventional trapping. Now, it's all well to inspect all these images, but sitting in front of a camera, sitting in front of a computer and going through hundreds of images every day to see if there's anything exotic shown there is also quite tedious and labor-intensive work. So can we not automate that? Yes, you can automate it by machine learning. And so the Fruit Fly IPM project is also looking into that matter, whether we can have a computer learning how to recognize an exotic or a target fruit fly from other fruit flies or from other insects which are being trapped inside your trap. And then if he finds something that is not supposed there, he raises the flag and he warns that this is something that needs a more detailed inspection. And the machine learning is like any other machine learning that most people are familiar with these days, and that is being used, for example, by naturalists to recognize plant species in the wild. You just take an image, and because of the repeated number of images that are coming in, you can actually train your computer to more easily recognize the target groups that you have. So actually these kind of traps have been developed, and at the moment we are deploying them. We are deploying them in two ways. One that you see here is an e-trap that has been established in the port of Thessaloniki in Greece, in order to see if any emerging fruit flies from consignments that escape from the containers can be detected that way. But we're also deploying these traps at the moment in pilot orchards in different countries, linked up with the modem system to see if this approach actually works as we want it to work. The third indication, or the third group, is identification of the intercepted and the trapped fruit flies. The machine learning is fine if you are talking about one or two different flies, but there are two shortcomings. If you have, for example, interceptions in the larval stage, most of the fruit fly larvae are quite non-descriptive. There are not many characters that you can use to recognize them. So they're quite uniform in shape. And even adult fruit flies are not always easy to differentiate one from another. What you see here are 9 fruit flies, which are 9 different species, but they all belong more or less to the same group, and they attack the same kind of fruits, or most of them do. So you can already see that it's not, for a non-specialist, it's not that easy to see the differences between these 9 flies. So the morphological and molecular identification tools that we are developing are in 3 groups. One of them is multi-entry keys for adults and larvae fruit flies. One of them is called a LAMP. And then linked to that one, and more linked to the molecular identification tools, we also try to establish whether we can use these tools not just to identify the species, but also to trace the origin of where the fly is coming from. So let me first talk about the multi-entry key. What is the principle? If you have an identification key in the normal way that most people are familiar with, it's what we call a dichotomous. The key takes you through a fixed pattern, and it will start with certain characters that you have to recognize on your organism. And for example, you can have a key and it will start with, are the legs yellow or black? Well, if you have a damaged fly and the legs are not really visible, you're stuck because you cannot tell whether they are yellow or black. A multi-entry key gives you a list of characters that you can choose from yourself, and then a list of entities, the number of species among which you want to identify. And so by having the choice of which characters, it's up to you what you choose. If the legs are not there, maybe the head is there, and you can look at characters of the head, or characters of the wing, or characters of the abdomen. So you can actually select what kind of characters you want to use. And by selecting, you will reduce the number of entities. So in the top left quarter here, you have the characters you can choose from. And for example, this one is about a yellow marking on the body of the fly, if it's present or absent. By saying it's present, you discard a number of entities. We started with 23. At 23, if you look at the top right corner, the entities remaining now are 3 species. The ones that are discarded are on the bottom right. So rather than having 23, you now have to choose from 3 species. So this way, you slowly, gradually reduce the number of entities until in the end, you end up with a single one. And so it's a much easier way to facilitate the identification by the non-specialist. The great advantage of these multi-entry keys in digital format is also that you can include a lot of pictures. You can include a lot of additional information for the user, because most people are not that familiar with all the different terms. Well, you can actually put images or a series of images to illustrate each of the characters that you want to earmark. You can use with arrows, you can use with circles to pinpoint which area people have to look for. And also because of the digital format, once you have made identification, like in this case, one entity remaining, you can actually link it to a lot of additional information. You can provide hyperlinks to other websites where you will find a more detailed description, where you will find a distribution map, where you will find molecular tools to identify them. So it is a very easy way for the non-specialist to be assisted in his identification. We developed this as a laptop tool, but also recently we converted it into a mobile application. So the FF-IPM project developed a mobile application for 23 target species, which were defined by a number of national plant protection organizations and by EPRO to be of importance for quarantine purposes within the European Union. And the key is freely available both for Android and Apple structured mobiles. You find the links at the bottom of this slide, but you can also find the links on the FF-IPM website. We have done that for the adults. We are now trying to see whether we can develop something similar for larvae. With larvae, it's much more difficult because, as I said already, the number of characters that you have in larvae is more restricted. It's not so broad. And also, it's not always easy to recognize one species or even a species group from another. So the larval key will mainly work with measurements, and it is actually more intended to support laboratory diagnostic services than actually something that you can do on hand in the field. But we still believe that it will facilitate the identification at this early stage. The second element that we are developing is a molecular tool. We already have molecular tools like DNA barcodes, but the fact is that if you want to do a DNA barcode, you actually need a fairly well-established molecular laboratory. And it's also a quite time-consuming approach. It takes, let's say, 1 day at least to get an answer. There is a technique which is called LAMP, that stands for loop-mediated isothermal amplification, which is a single tube technique for the amplification of DNA. It's at a low cost, it's sensitive and specific, and it goes much faster. It actually is based on the same system like DNA barcode. You have a specific genetic marker, but amplifying that genetic marker in the sample that you have is done at a very similar, at a very more reduced way than you do it for DNA barcodes. You don't need the building up of chains and breaking them down again at different temperatures. It can be done at one temperature. And you can actually, rather than having to read and to make a sequence of your results, because in a DNA barcode you will still provide a DNA sequence, you actually can have a color reaction because you can link the appropriate marker with illumination, and you can actually just visually see the absence or the presence of your DNA that you are earmarking. It goes also much faster. You can do it like in 1 hour. So it's a technique that can actually be applied or that can be implemented at a point of entry. You can actually establish this method at a port, at the airport or in harbor, and you don't need to send specimens and samples to a dedicated laboratory. Some lamps were already developed for a number of species, even within the target species that we have, but what we are mainly looking at is whether they are specific enough and whether we can develop lamps for species that are very closely related to these ones. And so there is one group that is looking at related species of Ceratitis, and there's another group that will be looking at the related species of Bactrocerra zonata and dorsalis. Molecular tools can also take us much further. Not with LAMP, but with other markers, we can actually try to develop a way of tracing the origin. While the species is one level, the populations is a different level. And we have, through a worldwide study, found out that actually populations in different parts of the world do not have the same characteristics. And so by selecting specific markers or parts of the DNA which are characteristic for certain populations, we could actually pinpoint the origin of certain flies. It will not be to a locality, but it will be to larger areas. What you see here, for example, is a study that was done on genetic markers, in this case, microsatellites of Ceratitis capitata from the area of origin, Africa, then the Mediterranean region, South America, Central America, and Australia. And actually you can see, for example, that you can, with the colored bars that you have here, that the Central American populations are actually different from the South American populations. So you could actually tell a difference whether the fly is coming from one part of the world or the other. The last thing I'm going to talk about is actually the prediction models. Fruit flies is one of the major groups of organisms that is being intercepted at the moment. We have about 30% of all interceptions in the EU are actually on fruit flies. So can we have any way of helping us in assisting us in what can we predict, on what kind of interceptions we can expect? Well, we can already look at what are the countries where most of the records are coming from, and you can find out that certain countries, the imports from those areas have a higher number of infested fruits. You have a seasonal thing. The number of flies or of interceptions is not the same throughout the whole year. It changes throughout the year, and it also changes according to the fruit species that is being introduced. Not every fruit is infested at the same number. And so we are, first of all, looking at the way of, can we actually have this kind of information combined into one prediction model of what kind of interceptions we can change. So we're trying to combine all these changes over time. And what you see here is actually 3 screenshots of a continuous time slot that analyzed the records of interceptions over time. And from left to right, you see a change from May, July, and December. And you actually can see that interceptions in the EU differ according to the regions and to the types. For example, in May, we will have a higher number of interceptions from Western Africa. It coincides with the mango season, which is then taken over in July by more interceptions from the Asian region. And then in December, you see that the number is lower, and that, for example, there's a slight increase in records from East Africa, because the mango season in East Africa is then in full swing. But there is not that much import of, for example, fruits like mango from East Africa in the EU. Most of it is coming from other regions. So by having this kind of predictive models regarding the interceptions, we can actually have a more targeted inspection done by at the points of entry. We can actually inform inspection officers in which time of the year they have to look out for which kind of interceptions from which regions and on which fruits. The second step on the predictive modeling is that, can we make any predictions on the suitability of certain regions within the country regarding their climate suitability over the year? If you have an interception from an area, what is the likelihood that your conditions locally are suitable for establishment? And here you can see one simulation for the area of the Adriatic Sea. What you see there is Croatia over time. And then if you go from January to August, you see an increase in suitability in different parts of that country throughout the period. However, I'm not going to talk too much about this topic. I will leave it here because it is actually, from what I understand, the topic of one of the forthcoming webinars, and I would not like to spoil all the surprises and the discoveries that other people will be able to present to you. So this more or less concludes my presentation. If you want to find out more about the FF-IPM project, the tools and the recent developments, there are different channels of information that you can consult. And I would like to thank you for your attention to this first webinar. Thank you very much.
Thank you very much, Marc. That was a really nice presentation and a nice overview also of some of the accomplishments of the project and some really interesting insights into this important topic on detection and on interception and on detection of fruit flies in Europe and elsewhere, because this was much broader, and it is much broader. So if there are questions, you can please write them, and then I can ask Marc to respond to these questions.
Or not. You have the answer.
I have actually a question for you, and this regards the identification of larvae. And the focus right now on the tools that you are working on is on the last instar, not the earlier ones. Is it, or would it be possible to morphologically, let's say, identify earlier instars?
It's actually, it's a good question, because indeed I forgot to mention that one, that the third larval instar is the main focus. At the moment, it seems to be nearly impossible to use first or second larval instars to identify morphologically. Why? Because some of the structures are not that well sclerotized. You have a much smaller scale. Of course, they're much smaller, so they're not even easy to find. And apparently, the structures that are there that you could use are even more uniform in first instars than in third instars. They change over time. They grow and they modify over time. So at the moment, we do not envisage to tackle the identification at an earlier stage than the third larval instar, which may present a certain limitation to the morphological identification, because yes, indeed, you have to wait till you have a fully grown instar. And for that purpose, I think the molecular tools will be much more suitable, because you can actually identify even an egg up to species level if you can extract the DNA from it, and that is possible. We have been doing that, well, our lab, not specifically for fruit flies, but we have been doing that for mosquitoes, for example. So if you can do it for a mosquito, you can certainly do it for a fruit fly.
Okay, thank you, Marc. So there is a question from Marc Schuche from Brisbane.
Is he still awake?
Marc sends greetings, and he says that it's a very interesting work. And then regarding the dorsalis lamp test, are you aiming for a dorsalis CC lamp test, or one of the complex group of target pest species of the complex? It
will be actually
a large complex, as you know, indeed, with very closely related species, so potentially very challenging. So
it certainly is. So yes, I think at the moment the lamp development is done by our colleagues in Stellenbosch University. We will not be able to do a lamp for the different species within that complex. So no, that is not the purpose. That is not going to be challenged. I think that's too large a challenge, so it will be for the complex as we recognize it today, and mainly with differentiation between other closely related species, but outside the complex.
Okay, there is also a comment from Polychronis Reboulakis. It says that excellent talk, Marc. Congratulations. The electronic nose has been tested with large consignments of fruits where odors from few infested fruits are expected to be diluted. So this is a question whether this has been tested.
At the moment, that is ongoing. So far, the work that has been done and that has been reported is actually about the recognition, the identification of the characteristic volatiles, and about the training of the e-nose apparatus. We still have to wait for the actual implication or application in the field when indeed you will have a container full of fresh fruits and a few of them are infested, whether he will still be able to detect and to differentiate those infested ones out of it. So that is not, we don't have any results on that one.
There is also a comment from Alice van Schauers-Müller. This is a really nice comment that after the fruit travel to Europe, the larvae will definitely not be in the first or second instance. So because it takes some time, so we are expecting to intercept, let's say, last instar of larvae and rather than younger ones. So
yes, that's correct. I mean, that's correct. And that's also why a lot of the inspections at the moment are done when they are not sure that they actually even rear the flies out. But again, because, you know, then already if you have a third instar larvae, the period is shorter, but still it takes a much longer time. So that is why we actually are targeting the third instars. Okay, that's all the questions that we had in the question and answer here. So if there are no additional questions, then we can probably give the micro to Georgia. And I would like to thank you, Marc, very much for this nice presentation, and also to mention that, as Georgia said earlier, the event is recorded and will be posted online, so additional people can attend, and you can always come in contact with Marc for additional discussion, also with myself and with the FF-IPM partners who are developing the different tools. So thank you very much, Marc. Thank you.
Thank you both. Thank you, Professor Papadopoulos. Thank you, Mr. Meyer, for this presentation. Thank you all for joining and participating to our webinar. And as I mentioned earlier, we are expecting to have more actions for you the next months. So please stay tuned. You can follow our social media, the project's social media and the website. And you can subscribe even for updates, news regarding the new webinars that we are about to have for you. So thank you very much for your participation. We hope that we have covered your questions. If you have anything else for us, so you can, of course, contact us via the channels we have available through our website and social media. And we are here to reply any questions that might arise from this webinar. We are really appreciate your participation, and we are happy to see you in the next actions that we will organize. Thank you very much for today.